Great. Well, thanks very much for inviting me to be here and for the really nice introduction. So as Sarah said, the title of my talk is Deciphering Climate Lake Linkages by Coupling Diatom Ecology with Lake Sediment Records. And I just thought I would give you the main messages of the talk now, just to help sort of orient you and yeah, give you the take-home message now. So what I'm hoping you'll gain an even deeper appreciation for today is that by understanding more about diatom ecology, this provides us with a great tool or set of tools to decipher climate lake linkages over longer timescales from lake sediments. And that diatoms can tell us a rich history about how climate forces lake ecosystem changes. But understanding diatom ecology really is the key, of course, to being able to do that. And so what I want to do today is just take you through a few different approaches that have been used not just by my lab group, but have been used by a number of different investigators over time to really deepen that understanding of diatom ecology and the links between diatom ecology and climate in an effort to really deepen what we can understand about climate lake linkages over time. So just to start off with, if we think about climate forcing of lake ecosystems, this is a very simplified diagram of climate forcing of lake ecosystems. So this is a figure that comes out of Williamson et al. 2009. And it basically takes you through, in an effort to sort of distill some of this complexity, takes you through the climate responses and forcing part of the diagram at the top. So thinking about things like different forms of radiation, air temperature, and precipitation. And then, linking that into what types of physical and chemical responses we see in lake ecosystems that are driven by climate. Again, this is a huge oversimplification, but it, is one way to start to organize your thinking. And we know, just looking at some of these physical and chemical lake features that are responsive to climate, these are the types of things that diatoms are sensitive to. And so these factors that change with climate forcing are factors that, we often see some pretty rapid changes in diatom communities and different types of diatom responses with those lake ecosystem changes. We also know, of course, that there are other drivers of change happening simultaneously with climate change. And, so we're in a multi-driver world where there's a number of different changes going on and shaping what we're seeing in aquatic ecosystems. And among the changes that we're seeing in aquatic ecosystems, there are a number of interactions as well that are going on that are not depicted in this diagram. So all of that is just to say that, as we're thinking about linking climate lake interactions with diatom ecology, the complexity and variability of climate lake linkages is one of the big challenges here, and something that requires some pretty extensive work. Some of the other challenges that come basically when we think about some of this sort of so-called standard quantitative approaches that we've been using to decipher diatom environment relationships over time. We know that a number of those quantitative approaches have, really faced a lot of questions in terms of how robust they are. And that, I think, has driven us to think about new approaches and also revisit some formerly used approaches as well. And finally, another challenge I'll just point out is that sometimes paleolimnology is a bit on the margins of limnology. And when I say that, I mean the degree to which limnological processes are integrated into paleolimnological interpretations can vary. And I would argue that, better integration of the two is going to yield deeper insights. So really keeping those limnological processes in mind as we're trying to understand some of these longer term climate lake linkages is a really important part of interpreting our paleo records. Just gonna pause for 1 second here because half of my screen is really not showing up right now. Let me see if I can. Okay, that's better. Okay. So as I mentioned earlier, what I want to do today is talk about some different approaches, some that come from my work and some that comes from the work of others, to improve our diatom-based inferences of climate lake linkages from sediment records. All three of these approaches really focus on strengthening ecological knowledge of taxa and expanding integration of our understanding of limnological processes into those inferences. And I'm going to really focus on some of the linkages between diatom ecology and physical features today. So kind of looking at this first tier of climate forced lake features. But, of course, understand that these physical changes drive and interact with chemical changes as well. And so that is always part of the interpretation too. So the three major types of approaches that I'll talk about today are, having a focus on seasonal diatom distribution patterns, target taxa or indicator taxa, and then also looking at habitat preference as one of the approaches. So we'll start off by taking a look at some of the seasonal diatom distribution patterns and this type of approach in trying to better understand diatom ecology. And of course, there's been a long history of work on phytoplankton seasonal succession. A lot of great lake ecologists who have done extensive work helping us to understand more broadly phytoplankton seasonal succession. And diatoms are certainly part of that body of work. But we've also seen some studies that specifically were focused on deciphering climate-driven changes in diatom assemblages and then linking those to what we see in sediment records. And so I'm talking, for example, about the work that Sebastian Interlandi and Sue Killam did in the Yellowstone area, and then Paula Noble's really nice work from the Western United States. One of those figures is shown here from Paula's work. And really trying to get at, the seasonal distribution of different diatom taxa and also their vertical distribution in the water column, and using that sort of gradient analysis to try to better understand the ecology of different species, and then linking that into what we see in lake sediments. What I want to focus on a little bit more right now is some studies that I think are really elegant, really nice design, and that is studies that couple sediment traps and varved lake sediments to explore some of the seasonal diatom responses and really get at some finer resolution interpretations. So when talking about varved sediments, these are more or less annually resolved sediments. So you can see these layers, and using sediment traps for this type of work is really great because being able to decipher what's going on at the seasonal level can be quite relevant to interpretations with these varved lake sediments. Sediment traps are also really nice if you're thinking about trying to understand what's going on during the spring and winter periods where it's not always that easy to get out and do some sampling. So some of the foundational work I would say in this area is the work from Platt Bradbury, 1988, on Elk Lake in Minnesota. And this was a study where he coupled sediment trap data from the springs of two contrasting climate years. So one was an early ice out year, one was a late ice out year. And he used that to interpret patterns in a varved lake core. And specifically, he found that Fragillaria crotonensis tended to do better in the late ice out year, and Stephanodiscus minutilis in the early ice out year. He linked this to what was happening in terms of turnover during the springtime in those two different types of years, and then related that to what that meant in terms of phosphorus supply to the diatoms, and then how that resulted in some of the changes in their community structure. And use that information then to infer what climate may have looked like over the past 350 years from the Elk Lake core. So a really nice pairing of the sediment trap work with what was going on in the sediment core. And we've seen this type of approach resurrected, I would say, recently. And again, some really nice work that is coupling these sediment traps with some sediment cores. And so this is Kino's work from 2016 from a lake in Germany. This is one lake that actually rarely freezes, although it did freeze during at least one of the years they were looking at. And this is one where, again, looked at some high resolution patterns in terms of what was happening with the diatom communities in those sediment traps. This one also involved some really nice modeling of lake conditions using some meteorological data as well. And ultimately, while they had some species level data presented in the paper, what they found most useful was to develop a silica deposition index to infer spring conditions from the sediment core data. So again, just a nice example of combining some of these approaches. And then I also just wanted to talk about Dominique Meyer's work as well from Sweden. And this was a lake that does freeze regularly. And again, we're looking at some coupled sediment trap data, in this case, 14 years of that data coupled with a varved lake core. And in this case, Dominique found some pretty large year-to-year variability in diatom succession. And she was able to link some of that with the springtime conditions, but also some with autumn conditions as well. So gathered a lot of really nice information about what was going on with each of these species, how they could indicate some different conditions in the lakes that were being forced by climate. One of the interesting and also somewhat unsettling conclusions from her work was that basically, when we look at a slice from a core, in this case, a varved core, so we think, maybe a full year of sediment, we often think of that slice integrating everything that's been going on over the year. And certainly it does, but what she was finding was that, clearly there were certain events or seasons that tended to contribute much more to the assemblage that was left behind than what we might expect. And so another consideration to keep in mind from some of this work, thinking about how much certain seasons may influence the total record that you're seeing. Okay, so that's an approach, again, involving this sort of seasonal perspective. And what I also wanted to talk about as another approach here is thinking about target taxa and using target taxa, focusing in on certain species that we think can be particularly insightful for a driver or set of drivers. And so sometimes we call these indicator taxa. And we identify these through work similar to what I was just talking about, some of these seasonal distribution patterns. We can also identify them through coherent patterns in lake sediment records. And that's what we're seeing in this case. This is some work from Small et al. 2005, looking at a number of different lakes. These are the sediment profiles from a number of different lakes around the Arctic, and seeing a sort of common pattern across these lakes, an increase in the Cyclotella sensulata taxa since 1850. And so it raises questions, well, what's driving this similar change across lakes? What is it about this group that seems to be responsive to something? So the focus when we're talking about target taxa and understanding diatom ecology, we're looking at taxa that appear to have a strong affinity with some key lake feature. And the focus then of the work is to try to verify, quantify, or elucidate what is at the root of that association. And so my group has actually done quite a bit of work on this group, the Cyclotella sensulata group, largely because of this great paper by Small et al. From 2005 that identified this broad pattern with the Cyclotella sensulata taxa. And so we did some work in the US Rockies, in a number of oligotrophic lakes, high elevation oligotrophic lakes, trying to understand what was driving the distribution of a suite of these taxa. And so we did a number of studies looking at distribution patterns across lakes, thinking about gradients like mixing depth as part of what we're seeing in terms of thermal structure, and also some different nutrient gradients like nitrate concentrations And we mapped out what the distribution patterns were like of several of these species along various gradients. We also conducted some small scale experiments. So these are experiments with a factorial design where you can take different factors and test them. In this case, we looked at light and nutrients, so two factors that are strongly affected by thermal structure changes. And between these two types of approaches, we basically deduced that thermal structure is an important factor for a number of these different Cyclotella sensulata taxa. And we found some differences you can see across these three different taxa in terms of what the optimal conditions seem to be in terms of thermal structure for these species. And, as we think about it, we want to think about how thermal structure is controlled in lake ecosystems and what are the factors then as we're trying to think about climate forcing of thermal structure and what that's telling us about these lakes. And we know that as we're getting into shallower mixed systems, we're looking at systems that, depending on the size of the lake, tends to have a decreasing wind strength and or changes in water clarity. So lower water clarity if we're getting into some shallower mixing. So as I said, this was based on studies that were looking across systems and then a series of small scale experiments. And with the finding that thermal structure was important, that's the kind of thing that you really want to try to test at the whole ecosystem scale if you can. You think about, something like thermal structure to make it a bit more realistic. If we could look at it at the whole ecosystem scale, would be more suitable. So again, thinking about the idea of target taxa and focusing on certain species, we decided to focus on Discostella stolidra with that shallowest mixing depth optimum that we see here. And we designed a whole lake experiment that would test the idea of whether Discostella stolidra is responsive to that shallow mixing. So this is an experiment that we did in the Arctic, and we used a hydraulic lift system to basically deepen the mixed layer of a whole lake ecosystem. And we chose two lakes that were basically very similar to each other in terms of their morphometry. They both had shallow thermal structure, so shallow stratification during the summertime. And importantly, both of them had abundant populations of Discostella stolidra. And we did this experiment where we monitored the two systems in 2013 with no change to either. And then in 2014, we manipulated what we termed the experimental lake. And so just looking at what happened to Discostella stolidra, this is on a log scale. We've got our control lake and the experimental lake. And basically you can see that as we deepen the mix layer in the experimental lake, the population of Discostella stolidra dropped. And in fact, the densities were about 40 times lower with that deeper mixing. So just another test to basically assess, what is the relationship here with thermal structure for some of these target species. We can take that information then and, as I said, try to use it to better understand some long-term patterns in climate lake linkages. So these are two sediment records. They're oriented maybe just a little differently. So we've got time on the x-axis here. And then we're looking at the percent relative abundance of Discostella stolidra or small Discostellas in these two lakes. These two lakes are located in the upper Midwest. So they're in the same region, generally speaking, and both in a region that has been warming. But we see different patterns in terms of what has happened with these small cyclotellas in these two lakes. And we think about the relationship with thermal structure. This would suggest that in Lake of the Woods, we've seen a shift to shallower mixing, and in Siskiwit, a shift to deeper mixing. And if we take a look at some of the weather station data from these two areas, we can see that warming air temperatures in the two areas have actually led to some differences in how wind strength is changing in these two areas. And we see that there's been decreasing wind strength in the Lake of the Woods area, which is consistent with an inferred shallower mixing. And we know there's been increasing wind strength in the region in which Siskiwit Lake is situated. And that's consistent with our inference here of deeper mixing over time. So just an example where, again, that deeper understanding of diatom ecology, is helping us to better understand some of these climate lake linkages and decipher what's going on here. We've been doing some work as well with Alacasyra, and this is work that Edna Pedraza-Garzon did as part of her PhD. And I think maybe I'm oversimplifying it, but I do think many of us tend to kind of lump, to some extent, Alacasyras together and think about them as, to some extent, having similar ecology to each other. But this is a sediment record from Beauty Lake in Montana. And I just wanted to show this different, the different Alacasyra species here, and over, 2,000 years, quite a bit of variability in terms of how they responded. So quite a bit of complexity there that we weren't quite expecting. And so we've been trying to work on exploring the ecology of this group a bit more. And so Edna did some really nice work here where she looked at the ecology of Alacasyra pusilla in these oligotrophic alpine lakes. And she saw that over the course of summer, where you have stratified lake conditions, in a less transparent lake, Alacasyra pusilla was actually blooming in the surface waters. So at the height of stratification, it's up near the surface here. And in the more transparent system, she saw quite a bit more variability in the vertical distribution of this species. And she did some nice experiments where she looked at light exposure and how the density of the species changed. And actually under the lowest light exposure, we saw a decline in the density of this species. So I think there's still a lot more work to do on Alacasyras. They're a big group, but I think Edna made a really nice start with this. And finally, I just want to mention with along the lines of target taxa here, one approach that I think is really nice, and I hope we see more of this, and that is coupling some of this work with lake ecosystem models. So this is some work that Brandy and Sherry did back in 2016. And you can see basically coupling some of the understanding of diatom ecology with lake ecosystem models to try to better understand what's happening during some different time periods. So, okay, I'll move on then to the last approach that I'll talk about, and that is thinking more about habitat preference of diatoms and using that to infer some lake ecosystem changes. And in particular, I'll talk about planktic to benthic ratios, so the P to B ratio. And, we often still tend to think about when you have a higher lake level, you should tend to see more planktic diatoms compared to benthic ones. And then with a lowered lake level, presumably you would get some increased benthic input. And that certainly is true sometimes, but we know that that's not always the case. And we know a lot of that now really from some great work that Jeffrey Stone did, modeling diatom habitats in a lot of different lake ecosystems. I think it's fairly safe to say that really, as you modify lake level and alter the amount of habitat, benthic habitat can expand or shrink depending on the basin slope. So generally speaking, planktic habitat area is always going to decrease, although depending on what's happened with the benthic habitat, that can affect the proportion of the plankton quite differently. So just suffice it to say that it's not a straightforward relationship, and keeping the lake bathymetry in mind is really important in trying to predict how that proportion can change with lake level changes. And so this is the work that Jeffrey did from Foy Lake, looking at some of the changes in elevation, so declining lake level as we move down the diagram. And you can see how planktic area changes. And you can also then see in orange here how the amount of benthic area is changing. And it's complex. It's not always a straightforward thing. And one of the really nice things about Jeffrey's models as well is that he integrated a photic depth as part of it as well. So you can modify that in these models. So depending on how the transparency of the lake might be changing, this can influence the proportion of planktic to benthic habitat too. And so Jeffrey used this to reconstruct lake level in Foy Lake. Again, you can see if you look here, this is benthic area to planktic area. And then we're looking at declining lake level as you move down the diagram. And you can see it's really at this point here in lake level where you have the maximum benthic area, well, relative to planktic area, or I guess the important part is less planktic area in that case. But then, from that, you can use that information to infer what was going on during some of these benthic peaks. So this is one example where with a declining lake level, you actually see, if you core in the deep zone, quite a difference in terms of the planktic to benthic ratio that you would expect. So with a higher lake level, coring here in the deep zone, you're also integrating all of this shallow area and the benthic habitat. With a couple meters drop, you can see how this deep basin becomes isolated and you lose a lot of that benthic area. So again, just thinking more about what happens with the habitat here and how that will shape what we see in the diatom sediment record, and then being able to use that for lake level reconstructions. Okay, and then finally, I'll just talk about one case where we combined a couple of these approaches. So this is some work that I did with Suzanne McGowan, Marion Yallop, and Adam Heathcote. And this was some work in West Greenland that we were doing as part of a large collaborative study. And from monitoring data, we had found that across a set of lakes in West Greenland from the early 1990s to post-2010, there was earlier ice out, and we were seeing a coherent decrease in lake water DOC that we think was increasing water transparency during this time. So we took a look at diatom assemblages that came from surface sediments that were collected in the early 1990s, and then another set of surface sediments that were collected post-2010. And we compared the diatom assemblages in the two. So that's what these two figures are showing you. And the first one, we're looking at the relative abundance of Discostella stolidra over time. And you can see that even though there was clearly warming that occurred in West Greenland from the 1990s to 2013, we know this from the instrumental record. Even though there's warming, we see this decline in Discostella stolidra across the 18 lakes that we sampled. And we also saw that the planktic to benthic ratio declined over the same time frame. And so what we infer from this is that that decrease in Discostella stolidra was the result of deeper mixing in these lakes over time. And we think a lot of that has to do with the decline in DOC that happened across systems during this period. And that increase in benthic taxa, we think, is a function of potentially of increased water clarity. Again, largely because of the changes in DOC. There was some work by Antonia Law looking at lake area changes over that same time frame, and the changes were heterogeneous, so no consistent change in one direction or the other. So this is why we think some of these changes in water clarity might be driving what we're seeing here. But again, just another example where we're combining a couple of these approaches in this case to try to better understand some of these linkages. Okay, so conclusions. So hopefully in my brief overview here of some of these different approaches, you've seen that some of the advances in quantifying and clarifying links between diatom ecology and climate-driven lake ecosystem changes. I think hopefully they're helping us to better understand some of those changes that are occurring. These approaches do rely on tighter integration of knowledge of limnological processes into our interpretations. I do think there's a lot of promise in integrating more lake modeling approaches, as well as automated sampling and sensors, like some of the things you saw with the sediment trap work. And I'll just say thanks to everyone whose work I cited, and to Jeffrey Stone for the slides of the planktic to benthic work. So that I will stop.
Awesome, Jasmine. Thank you very much. I have some questions, but first I'm going to go to Anna, who asked if you've ever followed up with one of your experimental lakes after you've stopped the intervention of mixing the water. Does the community return to the lake's previous state in the following years?
We have not, unfortunately. It would have been really nice to be able to do that. But the lake that we worked on, that set of lakes that we worked on, we accessed by helicopter. So part of the reason with that was in order to get the equipment in, we needed helicopter assistance anyways, but we wanted a lake that was as isolated as possible too. So unfortunately, no.
I have a question for you. So the dominant diatom in the summertime in Lake Superior is Discostella pseudostyliger, and I don't know if you would be counting them together or not. So maybe that's the first part of my question. The second question I have is, and maybe it's just understanding your study design a little bit better, but is it your sense that perhaps the thermocline is deepening and the density of Discostella is lower, but maybe the total biomass of Discostella is still the same? Does that make sense? The same amount of Discostella living in more space because you've deepened the thermocline.
Well, I think I would say we'd probably have to do more work to figure that out for sure. But I would say that we often do sample multiple depths when we do try to quantify like where are they located. In oligotrophic lakes, we sample anywhere from 3 to 8 different depths, just depending on the overall depth of the system, to try to get at, where in the water column are the taxa. So, but it's certainly possible we miss it too. I mean, that's because it's not, we're not sampling every inch.
Yeah.
But I would think that, well, yeah, so that's for the ecological side of things. I was going to say, I would think the sediments would reflect, but that's a separate story.
Yeah, I guess, I mean, okay, so is it your sense that there is simply less Discostella, or there's more space for the Discostella to live in, so it seems less dense?
I think there is less, and I base that on the multiple lines of evidence in terms of sampling multiple locations. I guess I would say in that last slide that I showed with Greenland, that was surface sediments. So it should integrate.
And after I've never had the pleasure of working in a varved lake, and I can only begin to guess how much integration has gone on in every sediment increment I look at from a core. But does knowing that these seasonal influences play maybe a big role in the community that you see in a sediment core increment, has it made you more cautiously approach your interpretations, or does it give more definition and say like, oh, hey, look, there was a flood year there, you can see it because there's this peak in the stratigraph or something?
Yeah, well, so I actually don't work in varved lakes either. Yeah, those were some other studies. I would love to do that. But so yeah, most of the lakes I work in do integrate a decent amount in each slice. But I would say that, yeah, I mean, knowing some of the, the potential for greater representation of certain seasons compared to others, I would say that makes me even more interested to learn about some of that finer scale detail. But also, I do think there, knowing that regardless will help your interpretation, even if you are integrating quite a bit more time.
Looks like Sarah has a question.
Yeah.
Yeah, thank you so much for your talk, Jasmine. And I was really delighted to see that you included Platt Bradbury's work because he was such a great, thought about seasonality. And in case anybody doesn't know, Platt Bradbury was a scientist who was the last student of G. Evelyn Hutchinson in that incredible lake ecological lineage. Okay, anyway, my question is, how do you, you've worked, Jeffrey and you have worked on those shifting between benthic and planktonic taxa in lakes that are very shallow or very transparent. Do you see particular benthic taxa that show up as being good markers of change?
Markers of climate related
Change or nutrient rate. There may be sort of more bathed in nutrients. So what are the benthic taxa informative? And
yeah.
So how?
Yeah. I'm afraid I'm probably not the person to ask about that one. I wish I knew more about benthics, but I don't. I do tend to focus more on the plankton and then that ratio of planktic to benthic. But so I don't know if anybody else is on who has more expertise with benthic taxa and some of the inferences there. Because it's a great question, and I wish I had an answer for you.
Yeah, I know John Small did some really interesting work with lake ice cover, and it was sort of more proposing that when the period of ice cover is longer, that also shifts, sort of the amount of moat area in a lake, and that would affect a benthos.
Yeah, which I think also assumes that there's not much going on under the ice in some ways. And we know more and more that's being challenged. And we're seeing more and more like the evidence that, oh, there's actually a lot going on under the ice, but that also depends on the quality of the ice and the amount of snow in that area, so those types of things too. But I think there's definitely quite a bit of work that could be done with benthics in this realm.
Thank you.
Yeah, we have a couple more questions, and I just want to throw in my 2 cents.
Good.
One thing that's confounding with benthic taxa is, most of our water quality observations are taken in the plankton, and so there are I think some difficulties right at the lake sediment interface where you might end up with anaerobic conditions and nutrients might be represented and available in different ways than they are in the water column. And I think it's just something that we have to be, I think it'd be very interesting to know and explore more, but what the diatoms living in on the surface of the mud might be experiencing very different conditions than those in a mixed lake or even certainly a stratified lake. But I think it's a cool area of research to pursue in the future for sure.
Yeah, absolutely.
Don Charles has a quick question. He asks if you have any experience trying to use diatoms and sediment cores to disentangle the effects of increased nutrient concentrations and climate change.
Yeah, I would say I do to some extent, and I think there are probably others who have worked on this as well. With my work, I mean, that's mostly been out west, looking at some of the effects of atmospheric nitrogen deposition and trying to also determine to what extent we're seeing a climate signal there. And I would say it's that has been challenging in terms of trying to isolate factors or try to think about, how do you quantify that interaction as well. We've done a little bit of that by trying to look at systems like glacially fed systems, where at west, that glacial meltwater increases the nutrient concentration of lakes and actually in some cases also keeps them cooler. So we try to look at some patterns in those systems as a contrast to see what can we infer as we do some comparisons across systems in that way. But I think there are some good gradients we can use to try to decipher some of that a bit more. But I haven't done a lot of that yet. So.
And Michaela has a question.
Thank you. Actually, I'm sorry for my voice. I am just going through a cold.
No worries.
Thank you. I wanted to recommend regarding benthic taxa importance in reflecting lake conditions. There is a paper that was published by Aline Philibert. She worked on this in the late 90s, early 2000s. And what she did is that she started to extract from the calibration set the benthic species one by one, and to look at the power of prediction of the overall calibration set. And she actually found that the benthic species were very important in predicting either nutrients or pH, or I don't remember exactly how many factors she looked at. But yes, that's a work that she did, and it was proving that they are important. That was a calibration set from Quebec.
Okay, thanks.
Yeah.
Michaela, can you write her name in the chat? Elaine Philibert, is that it? Since I don't know how to spell it. Thanks for that comment.
No problem.
I'm waiting to see the name. Oh, there we go. Oh no, I'm waiting to see the name. There we go. Yeah, thanks for bringing her to my attention. At least I had not heard of her work, so can't wait to look into it. And I don't know, Jasmine, if you see, maybe this is a little bit of what Don was alluding to. I know in some boreal systems, as the climate warms and we have increased liquid precipitation, there's increased like carbon transport to lakes, and transparency has been declining with DOC increases. Do you see co-occurring nutrients influxes in Greenland with increased temperatures, or
that's pretty hard to tease out, because certainly that DOM pool will certainly bring nitrogen and phosphorus with it. It's really unclear what's available in that pool. My hunch would be the phosphorus more than the nitrogen in terms of actually being bioavailable, given how diatoms can use organic forms of phosphorus, but trying to trace, what's actually coming in with that is pretty tough from a biogeochemical standpoint. So I am sure there are nutrients coming in with DOM or not coming in if we're seeing a decline in DOC like we are in Greenland.
oh, wow.
But yeah, quantifying that is definitely a whole project on its own. Yeah.
Go ahead, Sarah.
Yeah, I had another question about the cyclotella, and when you are seeing those shifts in relation to mixing depth, I missed on your plots if it's relative abundance of the composition. What's happening biomass wise in terms of those shift? Is that representing more or less, sort of amount of algal material with those shifts?
Yeah, I would say it's been variable from what I've seen. So for the sediment cores, yeah, I was showing percent relative abundance. From our experiments, we do measure biovolume, biomass, and I would say it's been variable in terms of the effects that we see there. So.
And then
But
how about, let's see, Mihaela had in her text question had also mentioned harmful algal bloom species. I mean, I don't imagine you have that much in Greenland, but in Northeast lakes, do you see any trade-off of blue-greens with diatoms in the mixed layer?
That's a great question. And I have a PhD student who's on right now, Avery Lamb, who is going to be looking at cyanohabs in Maine lakes in a climate context. And hopefully she'll be able to report on that in the next couple of years.
Excellent. I'll be looking for that, Avery.
Does anyone else have any more questions for Jasmine? I could probably talk to you all afternoon, but I don't want to take up all the time. And if nobody else has any questions, we can go ahead and get out of here early. Then I can go get the talk uploaded real quick. Well, thank you very much, Jasmine. I'm not seeing anything else in the chat or nobody else raising their hands. I'm sure folks can figure out how to get a hold of you if they have more questions for you.
Yeah.
Thanks so much, Jasmine. I really love your work. I think you're right on with understanding the seasonal succession, and I'm really pleased to see it.
Thanks for inviting me.
Yeah, thank you.
Appreciate it. Yeah, cool.
All right, well, thank you all very much for attending. I am going to ask one last question after I say goodbye to everybody. I was just going to ask, and it's something that we're trying to look more into as well. But obviously the winter ecology of diatoms. I'm wondering, how much of the wiggle and some of those oligoceras might be explained by some of that. And, as you said, there's ice thickness and the type of ice and snow cover and all these light limiting factors that can go into it. But are you guys embarking on any winter ecology, limnology?
Yeah, well, so Vendi, who I think is maybe still on here, has done some in West Greenland. And we've done a little bit here in Maine. I'm hoping we do some more. But yeah, I do think for sure when you're thinking about some of those species that we think are often blooming in the spring, trying to get a better handle on when those blooms are starting, how long are they lasting? What are the conditions under which they bloom? So again, kind of a little bit like Bradbury's work, really trying to get at those early ice out, late ice out conditions and which taxa tend to fare better under those different conditions. Yeah, I think it's an important season that we're definitely studying a lot more, but there's much more work to be done there.
Yeah, I always wonder when we go to retrieve the sediment traps in May, they've been there for 6-7 months. And it's like, well,
yeah,
what would they have looked like halfway through? And they're so bulky, I can't imagine trying to retrieve them through the ice. But
yeah, well, little known paper that I studied dynamics under winter ice cover for my master's degree. And like you said, Jasmine, it's very interesting in terms of the characteristics of the ice cover. And I could post the publication, but it's kind of the end. I don't know, maybe I'll.
I remember that paper. I
You
like it.
remember that?
I do.
You can
Yeah,
post it for
yeah,
That
me,
was an
Sarah.
old paper.
yeah, I like it. Yeah.